In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory.
Welcome everybody to JAX Tech Talk with the technical information scientists at the Jackson Laboratory. My name is Dolores, and my role is to serve the research community with education and practical solutions. JAX Tech Talk is a weekly 15-minute broadcast where we answer frequently asked questions about using mice for medical research. Today, I have Dr. Keya Ghosh with me to talk about best practices reducing bias when using mice on studies and increase the translatability of in vivo results into the clinic. This is an important topic since systematic bias often results in overstatement of drug efficacy.
Thanks, Dolores. So in this context, bias is any tendency that prevents impartial consideration of a question, which can lead to the skewing of your results. There are several major sources of bias in clinical research, as you can see from this slide, and these can occur during the phases of trial planning, implementation, or data analysis and publication. So let me see if I can get my pointer working here. Not so much. Anyway, some of these biases stem from our expectations for, let's say, rejecting the null hypothesis, as it were, and thus are easy to identify, at least in principle, while others might stem from more cryptic and embedded methodological flaws. Examining each subtype of bias shown on this slide is outside the scope of our talk today, but we refer you to this excellent and thoughtful review. For this talk, we'll focus on illustrating a few examples of different kinds of bias in animal research, as well as the relationship between bias and reproducibility. So it's important, of course, that the assays and the preclinical data are reliable such that they can be reproduced within and across technicians and laboratories, and most importantly, that this data is translatable to the clinic. Dolores, would you go over the elements of a well-conceived experimental design that aims to be reproducible?
Absolutely. So reproducible study designs take into consideration the application of many key principles that minimize as many environmental variables as possible, as well as eliminating potential bias. So what you see here in this slide is the pillars of reproducibility that are shown, and this was published by Stacy Rizzo and Jill Zimmerman in 2016. And you can see that reproducibility can be achieved by developing rigorous experimental design that takes into consideration choosing not only appropriate controls, also randomization, counterbalancing, blinding, and also, very important, using suitable sample sizes. And all of this should be also performed by highly trained technical staff with demonstrated proficiency at conducting every single assay that is part of the study design, as shown here on the slide. So we wanted to talk about several measures that are widely known to reduce the risk of bias. And one of them is randomization. So to do this right, test subjects should be randomly assigned to treatment groups. And if there's an experimental group, for example, and a control group, the animals need to be randomly assigned to either one. If you assign the healthiest mice to a treatment group, this can make the drug look a lot better than it is. The other big one is counterbalancing. When baseline testing is conducted so that a drug treatment will be compared relative to pre-treatment baselines, then for mice with, for example, similar performance levels, the body weights should be evenly distributed across treatment groups. It is very important not to bias high and low performance into a single group, for example.
That's right. And also, most of you know this, the fourth pillar here that we see, blinding, is very important as well. In an experiment, at a minimum, the staff responsible for directly evaluating, for instance, the effect of a treatment on an animal's health, whether that's looking at the phenotypes and collecting the data or analyzing that data, should not be aware of the treatment groups, i.e. which is the treatment, which is the control, or multiple study arms if you have them. The staff ideally should be blinded as to which animal underwent which procedure. The last pillar shown here is calculating the sample size need, which Dolores kind of alluded to earlier. And so a lot of times what you see is folks accumulating data continually until they find the result they're looking for. And that's another form of bias. So you really want to calculate the sample size based on your scientific needs and then go with the conclusion that you find after that.
Right, and you never can go wrong with consulting with a biostatistician even before starting the experiment. That is really what's very useful in my career. Just consult with them before you even get started to see what is the number of mice and other details for your research to avoid being biased. So, in fact, according to the literature, the main sources of bias when conducting research are lack of randomization and lack of being blinded to the assessment of an outcome. That is the slide that we posted right there. This is work by MacLeod and collaborators in 2015. When bias, whether you're conscious or unconscious about it, this can make candidate medical treatments look better than they are, like I said before. And this is a problem because they can also dampen apparent side effects when that's what you're being tested. So Science magazine published this chart that we're seeing right now, showing that 70% or more of 2,500 papers did not report analyzing the outcome in a blinded manner. And 75% of those papers did not report randomizing animals into groups, making it impossible for readers to evaluate the quality of the work. So when you're reading a paper like that, you just don't know if they blinded themselves but didn't report it, or they didn't do any of that to begin with. So researchers often ask us, what are the most common types of blinding in preclinical research, and how is this different from what we would see in clinical trials? Can you comment on that, Kaya?
Sure thing. So about the concept of blinding. In mouse studies, of course, the most important consideration is blinding the experimenters to the treatments that are being administered. For example, experimental versus control, as well as to the study subjects, which in the case of mouse experiments might be a transgenic or other mutant mouse compared to a wild type control or a littermate control. And this is an analogy what is done with human subjects and test agents in clinical trials. But in human trials, there are other layers of blinding, which you can see in this table, that are often employed, such as double blinding, double masked, etc. So while we wouldn't normally think of double blinding in a mouse study, there are exceptions, though, which we don't have time to get into. The last layer of triple blinding, i.e. blinding the data analysts to which data they're handling, and should also be considered in certain situations, especially when data point inclusion, exclusion routines are in place, or other complex multi-step data analysis is required. So we can see that there are, in fact, multiple parallels for blinding between animal and human studies.
That's great. Thank you for that. Another factor to reduce bias is avoiding potential conflicts of interest. Unfortunately, most of the times, this aspect is not reported either. I know a lot of search journals are having stricter guidelines with that. In the next section of this talk, we wanted to share a couple of examples of efficacy studies where the assessment of the phenotype can be a little bit subjective, even leaving even more room for bias. And those are tricky because without a strict training of the personnel conducting these studies, results may be different depending on who is collecting the data. So this is what is known as inter-rater variability. And also in many cases, using new technology, we'll see how this helps decrease this particular bias.
So if you're watching this episode live, feel free to type in your suggestions on how your lab or your research group works to decrease bias. that would be an interesting thing to discuss if we have time at the end. Whoops.
Perfect. That's the slide I wanted. So this first example is using mouse models of psoriasis, where grading of the severity of illness is important for the assessment of treatment efficacy. The problem is that most conventional assessments of psoriasis are subjective and have inevitable inter-observer variations or intra-observer variations as well. What you can see here in this table is how the scoring works. This is the PASI scores for mice. So within a given area of the body, the severity is estimated by 3 clinical signs: erythema, which is the redness, scaling, and decamation, or scaling or decamation, and the thickness of the area. And the scale goes from 0 of no alteration to 4, which is very distinct alteration. But what you can see there, that whether the phenotype is slight, moderate, or marked, sometimes that can be a blurry, distinction. However, psoriatic area assessment and severity assessments were found in many publications to be non-reproducible, and that is why maybe it depends on the intensity of the day, or depends on the experience of the person evaluating. This is very convenient in the sense that you don't need any instruments to do it, but it does have inherent subjectivity, which results in this variability that we were talking about. So how can we make this more reproducible if we are already designing experiments with all the pillars that we discussed? So the answer, go ahead.
I was just going to say that's a great question, Dolores. So, this is essentially qualitative data looking at things like degree of erythema. And so sometimes it's possible to take a qualitative data set and apply a semi-quantitative data analysis to it. And so you could contact, for instance, your image acquisition core. They're usually experts on both the hardware acquisition and the post-acquisition data analysis. And you can apply quantitative methods to analyze this data rather than subjectively scoring it. So the first thing you could do in a case like this is make sure that the ambient light in your lab is consistent from day to day when you're taking the pictures. Make sure your camera and microscope settings are exactly the same in terms of acquisition settings, right down to illumination, condenser, anything more specific. And then once you take the color images, you could consider separating them into component RGB channels. You could elect to choose, let's say, the red channel as the channel of chief interest, since we're talking about erythema. Convert that to black and white, look at pixel intensity in the channel, maybe mean or median. And that's, again, something your imaging core can consult on. And then compare that across test subjects rather than, looking at it and comparing it to some figure you might have found in the literature. And there's even more sophisticated ways to do it. You can use various plugins for color deconvolution in Fiji or ImageJ and other image processing software.
Excellent. Thank you so much for that. So you also had another example where readouts can be tightened to improve reproducibility. Can you share that with us as well?
Absolutely. Hang on a sec. I just having trouble changing this slide. Let me just, there we go. So basically, another great example here, and I apologize, I cannot erase these annotations, so just ignore those. But this is an example of EAE, or experimental autoimmune encephalitis. And that's a common test that's used obviously to study multiple sclerosis, but it's also used widely in the immunology field as a T-cell immune-mediated disease, or it's a model for any T-cell immune-mediated mechanism you might be studying. So in this case, this is a 5-point scoring system. It progresses from completely normal or lack of pathology down to various degrees of paralysis, hemiplegia, quadriplegia. And so this is already, you can see it's semi-quantitative already, but what you can do to reduce bias even further, and I really like this because it's a very obvious and simple thing to incorporate, is graded on 0.5 increments. So not just 1, 2, 3, 4, 5, but 1.5, 2.5. So an example might be, you can see that the Grading score 3 here is partial paralysis, where you're having one hind limb paralyzed. Maybe 2.5 would be that the hind limb is exhibiting a limp, but not completely paralyzed. So it sounds very simple, but what it does is it reduces the temptation to score higher or lower than the actual pathology that the mouse is exhibiting. Again, getting back to blinding, mice should be scored blind. This is a clinical scoring system that's widely used. It's very good, but it's also very subject to bias in whoever is collecting the data. You can also treat the mice blind, including the person not knowing what treatment is being administered to what cage, maybe not administering the treatment on a per cage basis, which is again something you might not want to do in the first place if you're using the counterbalancing and randomization procedures that Dolores mentioned, where you're sort of partitioning out the mice according to body weight so that it's evenly distributed across all your treatment groups, rather than it being on a per cage basis where you might have some imbalances there. And again, since these methods are somewhat subjective, I wanted to mention there's newer methods out there to collect data on mobility in these types of assays, and that involves motion detection sensors and video recordings within the cage. And so that basically takes the human interpreter out of the equation and removes quite a lot of bias.
Great, great. I wanted to take a minute here to address some of the questions that are coming in. We have a question from one of our viewers. What might the role of journals be in supporting randomization and bias reduction? That's a great question, because part of the problem is the journals, the reviewers should be asking for these things. And actually, most journals, scientific journals now, are more and more asking for this. What I suggest is that people refer to the ARRIVE guidelines. This is a consortium that was originated in the UK, but it's also has the support of researchers all over the world. And part of the ARRIVE guidelines is not only to, for example, spell out not only the strain that you're using, the stock number, the genetic background as an example for it to be reproducible, but also they are encouraging to do all of these very robust study design. And the idea is that a lot of journals, the reviewers are backing the ARRIVE guidelines, so that is something that is very encouraging for future research. So that what you're reading is something that was sound experiments and therefore sound results. I really like that question. Another question that came in is, can you give us an example of how JAX trains their techs to aim maximum reproducibility on preclinical studies? Sure. So basically, for any given test that we use in an efficacy study, let's say is a rotarod to look at motor coordination, then what we do is give our technicians groups of mice, and they are blinded to the groups of mice that we know that have problems, phenotype of problematic locomotion. Let's say it's an SOD1 ALS strain. And we also give them non-carrier controls, or wild types, if you will. And these are strains that are very well characterized. We know exactly the natural history, so we know how they would perform at any given age. So we give the technicians these two sets of mice. It could be even treated and untreated. And until they don't demonstrate that they can get the readings as expected, then we don't allocate those technicians, into actual efficacy studies. So there's a very rigorous training programs that have to be updated every year, and that is a way to make sure that technicians are actually reading the phenotypes with less bias as possible. Of course, they are. The mice are randomized and the technicians are blind to what they're getting. So that is just an example, but I mean, they are trained on every single assay in a similar way. Is until they cannot demonstrate what controls and the disease phenotype are supposed to do blinded, then they just cannot participate in further studies.
Thanks, Dolores. I think I saw one more question, which is how could you control for biases that you might not be aware of? And that's something that was shown in one of the earlier slides, that there are biases that are not obvious and you should still make an effort to control for them. So I actually have a really nice anecdote about that. So the first thing is just recognizing, that that's a problem. It's something you should be aware of. And I think it comes up even more often if you're performing a study in your lab that you're not accustomed to, if it's out of your wheelhouse, either methodologically or it's just a study that's maybe from another related discipline, but not your core discipline. And the example I have in mind is a COVID-19 mouse infection model, where researchers evaluated typical infectious disease readouts, which include things like survival, histopathology. But since it was COVID-19, they also wanted to evaluate anosmia, which is the loss of the sense of smell, which of course we also see in human infections. And so they used a social scent discrimination test to do that. And instead of just, finding the first study from the literature or, making up a way to do that, they went to an authoritative source. I believe it was current protocols in neuroscience, which is, if you think about it, an infectious disease researcher is not going to go very often to current protocols in neuroscience, right? But it's great that they did it because behavioral testing, again, it's generally a little bit out of the wheelhouse of an infectious disease researcher. So that article that they found, which, as we all know, like current protocols and other methodological journals, they're vetted by experts in the field. So the whole point of them is to give you the right method. So that article described this social scent discrimination test, which basically calculates the time to explore or locate the source of an interesting scent. And it compares that between, of course, infected and uninfected mice. But since infected mice are less mobile due to malaise, that's a confounding variable, which is actually a kind of bias. Confounding is a kind of bias. And so the article advised the researchers that you can, rather than just measuring the time directly and comparing it, you can calculate a preference index, which is essentially a ratio of the time spent interacting with interesting scent to the total time spent exploring an interesting scent and a control scent. So in this way, not only was the confounding variable addressed, but the confirmation bias of the researchers, where they might have a confirmation bias to accept that, oh, the mice are exhibiting anosmia just as human populations do, right? That's a great thing to put in your paper, but you need to control for that confirmation bias. So they're able to simultaneously at least, I won't say eliminate, but reduce both of those forms of biases by including some additional complexity into their experiment, which I think probably helped them in the peer review process as well.
Yeah, that's a great example, Kaya. Thank you so much. So that's all the time we have for this week. But before we go, check out the links in the description to find a webinar about achieving reproducible mouse studies, and a link to connect with our in vivo pharmacology teams if you need to run an efficacy study with our experts and do it the right way. So there's the two links for you to check. Thank you so much, Kaya, for joining us this week, and our viewers as well. Our next Tech Talk is episode number 58, Rewind. Let's talk about exciting JTT Q&As on August 16th. This is a video where we highlight the best questions from our viewers over the last 6 months. So check it out to see if your question is out there on August 16th. You can follow JAX Tech Talk on LinkedIn, so you're notified when we have a new episode, or you may also subscribe to our playlist on YouTube. This is Dolores saying, stay healthy, stay cool this summer, and stay excited about your research. Bye, everybody.
Bye.